Hello everyone watching this class. Today in the part 4 lecture of sexual reproduction in angiosperms, we are going to discuss megasporogenesis. So yesterday I had started with female reproductive system and today I will speak about megasporogenesis and further we will move towards the next topic also. So what is megasporogenesis? See, Megasporogenesis means megasporogenesis. This word actually means megaspore genesis means formation. So the process of formation of megaspore is known as megasporogenesis. Megasporogenesis is this is a process. Now how this process occurs? Basically, there is a diploid cell. This diploid cell is known as microspore mother cell. Oh, sorry. It is known as megaspore mother cell. Okay, this megaspore mother cell, since both are from MMM, like I had also taught in microsporogenesis, so that is microspore, and here it is megaspore mother cell. Now what will happen? This cell will produce megaspores. Megaspores. Okay, this cell will produce megaspores. And megaspores will be haploid. How will be the megaspores? Megaspores will be haploid. So this is like it will have like this megaspores. This will undergo again cell division, and after undergoing cell division, you will get number of cells. We will discuss this. Surely we are going to discuss. So let's speak about the details process. Okay. So I will write on the next slide. So I told that there is megaspore mother cell. So this cell is a diploid cell. Okay, this is that cell and this cell is diploid. So this cell undergoes a meiotic division. Okay, meiotic division, that is meiosis. Okay, so it undergoes meiosis. So we know that one cell in meiosis, out of one cell we get total 4 cells at the end of meiosis. If you don't know this concept, then go and watch my lecture, Introduction to Cell Division. There I have taught the basics. You will understand it within 50 minutes. 50 minutes. Go and like watch it once. So there are 4 cells are formed, and these each cell is haploid. Okay, so here there is a formation of 4 haploid megaspores. Okay, 4 haploid megaspores. Okay, so after this what happens? See, there will be degeneration. Okay, the out of these 4 cells, 3 cells will get degenerated. So the cells which is towards micropyle, okay, the cells which are towards micropyle region will get degenerated, and one cell will remain in the in which region, at the chalazal region. Okay, so at the chalazar region one cell will remain. So you can show it. Now here we can't say where is chalazar end, because here we have not taken any reference. But suppose if I take reference that upper side is micropylar region and lower side is chalazar region, so we can draw here one megaspore. Okay, so out of these four megaspore, one megaspore is like only one megaspore is present. Rest three megaspores are degenerated. Okay, let's begin. Let's go forward. So now what will happen? See, there is only one cell like this only one megaspore. So I will speak about megaspore here. Now suppose this is megaspore which you have taken. Just taken a zoomed version of this cell. If you can see, I will mark it. This cell. We have zoomed it here. Okay, we are just zooming it. And now we will see that how many cell division this megaspore will undergo. So there will be the very first cell division, okay, that is known as mitosis. Now we know that in mitosis, if there is haploid cell, okay, if this is a haploid cell, you will get again a haploid cell. But here what is happening, there is a free nuclear division. Means what? In one cell only, all the nucleus will divide. Okay, so this is one cell, one megaspore, and here you will get two nucleus. How many nucleus? Two nucleus first in the first first time mitosis has occurred. First time. Okay, first time mitosis. Now if suppose there is again second time mitosis occurring, again there will be nuclear division. So here you will get how many nucleus? You will get four nucleus, and all will be haploid. Okay, all will be haploid. So you can see that this is mitosis number 1. Mitosis number 2 means second time mitosis has occurred. For how many times it is occurring? If I move forward, suppose now after this there is third mitosis occurring. So I will write here mitosis again occurring here. So you will again have this um, cell division. You will have total 4 cells, like 4 up 4 down, 8 cells. So in short you can say this one is like third time mitosis. So it is 2 raised to n, where n stands for number of mitosis. So here mitosis has occurred 3 times. So 2 into 2 into like 3 times if you do, then you get 8 cells. Now here 8 nucleus you can say. But this nucleus will get its own like covering also. Means each nucleus will get its own cell wall. Here we will say cell wall. Okay, so here what will happen if I speak about this like stigma, style and ovary. See, so this is stigma, this is your style, and this is ovary. Okay, here in the ovary we first had what? This is the embryo sac. Okay, that's what you can say, the ovary. Then you have the embryo sac, and inside embryo sac what you have, inside this embryo sac, or we can say this is ovule, and you have inside embryo sac. Okay, so you can say ovary, ovule, then you can say embryo sac. Okay, and inside this embryo sac you have what? You have megaspore cell. Okay, megaspore cell. So megaspore cell means which cell? This cell. Okay, this is megaspore, this megaspore, and this megaspore has completely divided into what you can see here. So now this will, now how this will be arranged, these 8 cells. See, this is your embryo sac. So that one megaspore which was like this, it has divided into 8 cells now. So 4 cells are going upward. Okay, this is what we saw in the last division. But see, this is very congested. You can't sit in one bench. In one bench you can't sit, 4 people can't sit. Okay, so what will happen? In middle there is a space. So this one nucleus, like one cell, and this one cell is coming in the center. Okay, they are coming in the center. So here you will have 3 cells, and here you will have 3 cells, and in between you will have 2 cells. The two cells will come, and they will fuse, but their nucleus won't fuse. The 2 cells which have came from the 2 poles. Okay, now this was a pole, this is one pole, and this is one pole. Here there were total 4 cells. Here there were total 4 cells. So one cell from here, okay, one cell from here, and one cell from here, they came into like in the middle. So here what happened, the cells fused, but their nucleus didn't fused. So here you will have 2 nuclei. I will draw a zoomed structure. Suppose this is the only thing which you are seeing, that is the embryo sac. Now, so I told upwards means which end? Upwards, I have taught yesterday that chalazon end is the above end, and the below where there is opening, then like small opening, there we say it as micropyle, or you can say micropylar end. Okay, so here you have 3 cells, here you have three three cells, and the middle one I have drawn bigger because that is egg, and here I have to draw this one cell with 2 nuclei. So total we are having how many cells? Total we are having 3 plus 3 plus 1. We are having 7 cells. And how many nucleus? We have 1, 2, 3. See here. This is 1, 2, 3, 4, 5, 6, 7, 8. We have 8 nucleus. So 8 nuclei. 8 nuclei. So this structure is known as 7-celled. So we say this structure as 7-celled 8 nuclear nucleic structure. Okay, nuclei, you can say 8 nuclei structure. So this is a very important thing. You should know that which type of structure is this. It is 7-celled 8 nuclei structure. Okay, above cells, these above cells, 3 cells, okay, those are known as antipodals. And these all cells are what? Haploid. This is haploid, haploid, haploid. This one nucleus haploid, those 2 nucleus, okay, haploid, haploid, haploid. All cells here are haploid. The middle cell will have n plus n, which you can say as 2n here as a whole because it's just one cell. But the nucleus have not fused. The middle cell we call it as secondary nucleus. Secondary nucleus. And down you have 2 synergids. synergids, and the middle one is known as egg. So two synergids plus egg, this entirely we call it as egg apparatus. Egg apparatus. Okay. And then here we also have like small finger like structure here also and here also, like it looks like this. So this structure which I have shown here, that is known as filiform apparatus. Okay, this you should know, filiform apparatus. Okay, so this is all about the part which we should know about um, Megasporogenesis. Now, if you can see that here only one megaspore was functional. You can also say megaspore here as functional megaspore. So there, since there was only one megaspore functional, so we say this type of, development as what we can say this type of development as monosporic development, because out of one spore entire embryo sac got developed. So we say monosporic development. You should know this, that it is monosporic development. This is the first point you should know, the very first point. Now the second point you should know that here the development is internal. Everything is happening inside. If you can see, everything is happening inside ovary, inside ovule, inside embryo sac. So it's happening inside. Since it's happening inside, so we say it as endosporous. Okay, so development is endosporous within the megaspore, and that megaspore is also present inside the embryo sac. Embryo sac is present inside the ovule. Ovule is present inside the ovary. You should know this. So the development here is endosporous, inside the spore. Endosporous development. Okay, so these two words you should know. And the last thing which I already mentioned, but I will write it here, the third point you should know is that the structure is seven celled, eight nuclei, embryo sac structure. Okay, embryo sac structure. Yes, so this is thing, and everything is concealed. Now what is concealed means, ovary, inside ovary you have ovule, inside ovule you have embryo sac, means it's everything is covered. Okay, it's concealed, it's present, one thing is present inside the under another thing. So everything is enclosed inside the ovary, right? So we say concealed structure. Embryo sac, the another word if you want for embryo sac, you can say another word as female gametophyte. What you can say? Female gametophyte. This word yesterday also I had given. Female gametophyte. Okay, so this is all. Here you can also say, instead of saying four, okay, like I will mention it because yesterday also when we were studying the pollen grains development microspore, there also I had told tetrad. You can write here tetrad also, tetrad haploid microspores, megaspores, sorry. So you can say here also tetrad instead of writing four word. Okay, you should remember this point also. So that's it. This was the entire topic about the development of embryo sac. Okay, and I told that development is this functional megaspore is towards chalazal end. Which end? C means chalazal end, and the three are degenerating. These are degenerating towards micropylar end. Okay, so this is all about the Megasporogenesis. So I one scientist name is left. Okay, I will tell you the name of the scientist also. See, this structure which we had told, like this structure which I have written name here also. So this structure was given by, like it was discovered by S.G. Navashin. You should know it. The name of the scientist. Even the double fertilization topic was given by S.G. Nava Navashin. You should know this. He has given embryo sac development. This also topic he has given. So he discovered this embryo sac, uh, like it is seven-celled, eight nuclei structure in a plant known as polygonum. Okay, in this particular types of plants, he discovered this structure of embryo sac. Okay, so this is entire thing which you should know about it. Next, further, we will speak about 3 points here. I will say. See, you should know that what converts to what. See, in future, ovule will convert into ovule will convert into seed. Okay, this is the very first point. Second point, ovary will convert into fruit. Okay, third point, if you can see the like yesterday I had taught about integuments and all, integuments, nucellus. So if integuments are there, and integuments also I told that integuments are like inner and outer, two layers are there, two coverings are there, integuments. So the outer integument and the inner, so outer and inner, these two layers converts into seed coat. Okay, what it converts into seed coats, seed coats. And outer integument, like outer integument is known as testa in seed coat. Okay, and inner integument, which is an inner seed coat of seed, that is known as tegment. So you should know these all conversions. Okay, the fourth point if I take, if suppose nucellus, now yesterday I had told what is nucellus. See, in this structure, where is anatropous, this is b1 is anatropous. You can see that this light color, okay, like inside, you see the light yellow color inside this embryo sac. We have nucellus. So nucellus is nutritive in function, it provides nutrition. So nucellus, if suppose nucellus is persistent, okay, means it's present for, like after fertilization also it's present, in the post fertilization it is present with the seed and fruit, then you can say that persistent nucellus as perisperm. What we can say, perisperm. So these are the four conversions. You can write fifth conversion as megaspore mother cell got converted into megaspore. Megaspore, and then this megaspore got converted into embryo sac. Okay, and this embryo sac has egg. Okay, it also has antipodals. It also has synergids. But the very important thing that it's having is egg. Okay, so you should know this much. These are the conversions for today. Next, I was showing you this image. See, in this image, I have shown that there are 6 types of ovule. Yesterday, I mentioned about anatropous ovule. So, this is anatropous ovule C1. See if you can see that the micropyle and funicle are adjacent. Okay, and micropyle is downward. See here, if you can see, the micropyle is upward. Yes, in second B diagram, you can see Sericinotropous ovule. If you can see, micropyle is upward, and here you can see that micropyle. Okay, the micropyle is upward with chalaza, like they are present closely, like very close to each other. Then micropyle, here you have chalaza and funicle. Okay, so these three are present in the same line, so we say it as orthotropous ovule. What we say? We say it as orthotropous ovule. Now if you can see that this the direction is thisward, like in hemianatropous, you can see that micropyle and funicle, they are somewhat in 90-degree angle, they are forming 90-degree angle, so we say it as hemianatropous. Then you have amphianatropous, like it is like it's curved structure. Even the embryo sac, you can see that it's curved. In camile campylotropous, you can see that again there is a curvedness. Okay, so these are the seven types. If you can see, these are the seven types of. Okay, I will erase these things, or I can put the diagram, one clean diagram. You should be able to, able to at least you should know ki ha, there are six types. You should know this much. Okay, this is a diagram which I drew here. Like I make you understand. I tried to explain you the six types. You can google these definitions and all, but just you should know that okay, there are six types. So this is a very good picture which I got from Google, in Britannica website. So you can see here that how they have explained entire flower, then how they are showing the male and female parts stigma, that the pollen grain is coming on it, and then pollen tube is like actually going inside. Where it's going in the ovule. See, this is one ovule, two ovule, three ovule, four ovule, five. There are six ovules here. There are many ovules, so multi-ovulate condition is here. Then you can also see here that egg cell is here green color. Besides it, you have two synergids. You can see polar nuclei. You can see here, uh, say this antipodals. Actually, here only three antipodals should come. I don't know why four is shown. Okay. Then you can see zygote formation. This we will study. From here we will study. This entire part is left, but you should be able to understand till here. Okay, you should be able to understand till here. Fine. So this was about today's class, and this is a diagram of again female gametophyte. But here if you can see that the pollen grains have came here, like reached on the stigma, and they are like this pollen grain has formed what? Pollen tube. See, it's written here, it has formed pollen tube, and it's going towards, you know it's going towards the micropylar end, and it's going there to female. Germ cell is nothing but egg. So this is a good diagram which is taken from NCRT, and this is all about today's class. Now next we will come towards it, like we will extend some more time, like 10 minutes more. See, today's like next topic is pollination. So pollination, like what is pollination and why it is necessary. Okay, so if you can see here that the bee is sitting on this, you can see here that bee is sitting on, honeybee is sitting on a sunflower. There are many insects going towards the flower and all. So what they are doing actually? See, you know the basic definition actually of this, but let's start from the basics. See, now what is pollination? So pollination is nothing but, it is like, what is the meaning of pollination? Pollination means what? Transfer of pollen grains, pollen grains from anther to stigma. Anther to stigma means from male to female, you can see. Now this pollen grains I have taught yesterday about microsporogenesis. This is actually having male gametes. But why there is why there is requirement of pollination? See, pollen grains, okay, pollen grains and male gametes both are non-motile. Yesterday I have mentioned both are non-motile. If suppose both are non-motile, then how they will reach to female? Okay, if both are non-motile, then is there any way they are, like, they are not having any capacity to fly and go? Okay, so for that purpose they need the process of pollination. But for doing the work of pollination they need some agent. Okay, there should be one agent which can move from one flower. Okay, see, this is a pollinator. That agent is actually known as a pollinator. That, see, now this honeybee first came to the flower number 1, and then the honeybee went to the flower number 2. So this is pollinator, means this is one agent. This agent is helping to carry pollen. See, she has carried her one pollen seed, carried, and she will release on this stigma. See, the pollens have reached here and it will fall on the stigma. So the process of taking pollens from one flower to another, okay, that is known as pollination. And the work, okay, the person who is working, who is helping in carrying the pollen and delivery of the pollen, okay, now what this pollinator will do, it will carry and deliver the pollen, like the delivery boy, the person who brings the parcel, person who brings the food from one place, like from hotel to your house, okay, or from one hotel to another hotel, whatever, or from one company to your home, whatever. So that person who is carrying and delivering the thing is known as agent. In this proper language we say it as, in a fashionable language you can say, we say it as pollinator. It's carrying pollen and the process is known as pollination. I hope you understand that the three words, okay, the very important three words, that what is pollination, why pollination is needed, and who is doing pollination. So I hope you understood these three words. Okay, you can see that honeybees, actually honeybees are the major pollinators. Okay, the maximum pollination is done by honeybees. Okay, maximum number of plants are pollinated by honeybees. So you should know. Okay, now coming to the next part, that is self and cross pollination. This I feel to start in the next lecture. So in upcoming class, I will teach you this, the self and all, in detail. We will speak about the pollination in much detail in upcoming class. And till then, keep studying, keep learning, and take care. Thank you for watching this class.